Good afternoon. I would like first to thank very much the organizers for inviting me just to share with you some thoughts about pediatric oncology, precision medicine. I will not talk about neuroblastoma. I will just discuss with you some challenges and some pitfalls. Yesterday, I tried to find a definition of precision medicine. I found this one on the NIH website. An emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person, in contrast to a one-size-fits-all. I think interesting because it's rather general. It gives a message it's not only in oncology, it's not only gene, it's beyond, but maybe it's too general. So I prefer the second one, which is the one from Joe Handelsman from the Precision Medicine Initiative in 2016 that I found in the internet as well. It's the use of cutting-edge technologies to match patients with the treatment that will make them well. It's simple, it's focused, and I think it means what it means. It means that we need to know biology to really use the cutting edge and treat better the patients. And regarding biology, clearly for me, you experts in neuroblastoma, for years and decades, you have been doing precision medicine. You have been looking at the biology of the tumor of the patients, and you have been using this information to improve the treatment, stratify the treatment. So clearly, this is an excellent example of the use of the information from the tumor of the patient to improve the treatment that we provide them with. But clearly, the targets are difficult. And this is a quote by Richard Gilbertson last February in London at the Sean Conference. And he was clearly saying that we have very good target. NMIC is an excellent target. And I'm sure that you are still frustrated as neuroblastoma experts not having a drug to target this hallmark of neuroblastoma. And clearly, this is one of the issues of specific drug development in the pediatric malignancies. Those specific targets are not easy to drug. But in addition, we have now a massive information about molecular biology, whole exome sequencing, RNA sequencing of pediatric malignancies in neuroblastoma, in brain tumors. And there's this most recent paper in Nature by Grobner looking at 1,000 tumors of different pediatric malignancies and giving the information about, for example, the mutation rate. But if you look at the conclusion or at the abstract, it says 7 to 8% have an unambiguous predisposition gene variance. It means that there may be more, but this is what we know at the moment. But more interestingly, it said 50%, 1 in 2 of these tumors, of this patient, have a potentially drug event. 1 in 2. That is extremely important. It means that if we really have druggable events, druggable target, and we have the drug, we can really make a difference. And we have paradigmatic example. And the paradigmatic example of precision medicine is, of course, larotrectinib. Larotrectinib for NTRK fusion tumors. And you know this data in the phase 1 in children with NTRK fusion tumors. All patients with the biomarker responded to larotrectinib, including this should be the biomarker, including this 2-year-old girl with a massive infantile fibrosarcoma and a rapid tumor response with the drug. So this is a paradigmatic example of precision medicine. An alteration which is strongly oncogenic driver and a good drug, and then we have really a very nice response and an activity. But not all the alteration we find are so driving at this one. And the question is, how can we get the information from the tumor of the patient to develop the best drugs we can just to match them, to match this alteration, and have a chance to have effects as this one probably. And at the moment, if we look at what's happening in the field, clearly there are commercially available tumor molecular portraits. It's really a huge activity, especially in the adult oncology world, because clearly when a patient has lung cancer, you need to know if there is an EGFR mutation, a RAS translocation, or KRAS, and so on, just to give him the drug that have been approved with biomarker. So clearly this activity is up and running and expanding. And clearly this is also available for pediatric patients. And what it is sending to these companies a tumor, frozen or FFPE. They do most of the time gene panel sequencing. They issue a report, and the report includes what the alteration found and some information about the alterations. What are the drugs known to target these alterations as a list? And in addition, in most of these portraits, what is available in terms of clinical trials. So clearly it means that for any patient, a physician can prescribe a portrait, get the information, and then propose a treatment. And this is an issue that we need to address, because prescribing off-label in such a situation without getting all the information is really an issue that we should address, because for me, precision medicine at the moment, it's still a research topic under evaluation for children and adolescents with cancer. We are not in the setting of lung cancer. We are still needing to generate information to do best for children, to develop new drugs, and to demonstrate that we can have these drugs as effective as they could be in adults. So clearly, precision medicine, what it is, this is first to generate molecular information from a tumor sample or liquid biopsy in a clinical setting, which means that it is not a research lab doing the sequencing usual. No, it's in a clinical setting with all the qualification of generating molecular information for a diagnosis. The second point is that there is a need to interpret. Is alteration found a driver or not a driver? Is it actionable or not actionable? And this is a magic word which is extremely complicated, because to really define what is actionable is difficult, and there is no clear definition from everywhere. Then to decide on what would be the matching treatment as a single agent or even more in combination, because we know that single agent is unlikely, most of the case, to be effective in those patients. Then the patient needs to be treated in a safe and secure environment, which is the framework of this should be a clinical trial or a capacity to generate the information safely and to collect this information. Because we need to collect the information. We need to validate. It's not only giving the drug and hoping it works. We need to validate in order to define whether or not this treatment in this population is relevant. And finally, to learn and discover, because generating all this information is very important just to improve further the way we develop this drug for children and we provide them with better treatment. So it's definitely a multidisciplinary experience. It's not a single physician prescribing a portrait and then a drug. It's really a multidisciplinary experience in a research environment, which I think is the only way to move forward at the moment. So in ITCC in Europe, we build a strategy, and the strategy is very simple. First, generate at relapse, biopsying the patients at relapse, a molecular and immunological portrait through molecular matching trials, doing whole exome sequencing, RNA sequencing, and trying to do some immunophenotyping. And then trying to match this alteration with a portfolio of phase 1 and phase 2 trial that we do have, single agent or combination, targeting agent or immunotherapy, just trying to match the alteration we find in the tumor patient and we can consider as potential target of the treatment we do have in phase 1 or in phase 2. And clearly the goal is that all patients entering the program is proposed to have access to innovative drugs. So it's different from the pediatric match. In the pediatric match, the patients enter the program, have the sequencing, then get the treatment within the pediatric match, and if not, get out of the pediatric match and get treatment outside. Here we have on one side the molecular profiling. On the other side, we have the portfolio of phase 1 and phase 2. And finally, the goal is to aggregate all this information and generate a clinical biological database from these tumors, from these patients exposed to these drugs, just to generate new knowledge, just to give the capacity to researcher, basic and translational researcher, to use this information and identify new pathway, new target for potential new drug development. So this is a strategy that we implemented 3 years ago, and it is up and running. It's someone running in France with the MAPIAX program. What it is, very simple. Patients and parents are consenting to enter this program, which is led by Birgit Dioger and Gudrun Schreier-Meyer. The goal is biopsy at relapse, whole exome and RNA sequencing, molecular portrait, molecular report with the biologist and the discussion, and then treatment of proposed in the phase 1 and phase 2. The goal is 600 children in 3 years. We started in January 2016, and at the moment we have 440 patients entering the trial. It's running in France, it's running in Italy, and very soon in Spain and in the Netherlands. In Germany, in the Heidelberg team with Olaf Witt and Stefan Pfister, the INFORM trial is running, is proposing at the time of relapse, whole exome sequencing, methylome and RNA sequencing, generating molecular portrait, can even think to the pediatric oncologist just to propose a treatment. So very much the same concept. And this program is running in Germany, it's running also in some limitrof countries, and clearly this is expanding in order to provide across Europe molecular profiling at relapse. But this is embedded in a program where clearly in the ITCC we have the INFORM in Germany, the MAPIAX in France, the SMPED is clearly up and running very soon in the UK, doing the same thing, molecular profiling at relapse, and the ITER program in the Netherlands. And what we will be doing from the national program, with harmonization of the pipelines, harmonization of the data, and bioinformaticians working together, we will create this unique aggregated clinical biological research database. And we target 3,000 exome by 2019. So as you see, it's generating information. We don't want to demonstrate that doing genomic medicine is better than doing chemotherapy. We want to generate information to develop new drugs. Main messages, different disease, mainly solid tumors. The main pathway altered are cell cycle, receptor tyrosine kinase, and apoptosis, and this is very much the same in the different program running in the same way. Interestingly, we found out that in most of these tumors, there is not only one potentially actionable target, but sometimes more than one, up to 3, which means that interpreting this molecular alteration is very difficult. And the way to define that this alteration is driver and not is not simple. And clearly, the discussions are very important just to find out what would be the best alteration to target. But coming to this point is saying that generating information for the patient is important, but what matters in precision oncology is access to medicine, rapid access to medicine. And this is where we need innovative design and trial platform to be sure we can propose to this patient access to innovation that would match the alteration they have. But we know that in 1 out of 2 at least, we will not have any matching drugs, but then we need to propose them as well access to innovation. So what are we doing? Clearly, the platform trials are very important. I think it's brand new. It's developing in the pediatric setting, it's developing as well in the adult setting. The concept is very simple. Several therapeutic arms in one trial for a given population, prospectively amended to including new treatment arms, which means that it's a capacity to enlarge as soon as you do have new information, but also to speed up the implementation. It's more rapid to amend a protocol than to start from scratch a new protocol. And this is very much, we think, very important just to have an acceleration of the capacity to develop this innovation for the patients. So what we did in ITCC, we considered that from the molecular profiling program that I mentioned, there is a portfolio of phase 1 and phase 2, but it did not cover all the alteration we found in the tumors of the patients. So what we did, we developed the ISMAT program, which is a trial platform, a platform trial. I never know which way it should be done, but clearly this is a trial with several therapeutic arms. The goal is to have more than 10 drugs from more than 3 companies as single agent and combo, and it was launched in 2016. So what it is, each arm is run independently. There are 2 parts, phase 1, dose escalation, and then expansion with a statistical design I will not go into, but it's very smart. These are phase 1 patients. We are looking for evaluation of safety and activity at the same time. And in addition, we enrich, when I go back. How can I go back? I did go back. And we enrich each arm with patients with a definitive profile, which is important. We are not only having a BRAF inhibitor for a BRAF mutation. And for most of the arm, there may be potential biomarker, but we don't know which biomarker is really the biomarker of a given drug. So by enriching, having patients with a profile that could fit and a profile that don't fit at all, we will generate information in this cohort of patients receiving new drugs just to find out, if possible, what are the biomarkers leading to activities or resistance. So clearly this is up and running, and in April we had 92 patients enrolled over 20 months, and the capacity in this trial to recruit twice the same patient, because clearly some patient can benefit from several arms. So we have 8 patients who receive different arms, no, only 2 in this trial. So this is phase 1 patients in the relapse setting who do have a deep tumor molecular analysis available, because we will have at the end all patients with this molecular data available in the program. So we started with 7 therapeutic arms, a CDK4/6, a WEE1 inhibitor, a PARP inhibitor, mTORC1 and 2, and PD-1. Most of them were enriched, not the nivolumab, because there is no way to enrich at that time a nivolumab trial. And we amended after 18 months, so it was quite rapid, with 3 new arms, a MEK-mTOR inhibitor combination, an IDH2 inhibitors, and a PD-1 and KIR inhibitors. And we are working with 4 different pharmas, and clearly we have different investigators for each of the different arms. So clearly it works as a platform. We could amend and open 3 new arms in a rapid way because it was amending a protocol already existing. The other example I wanted to give you is not only in this situation of the relapse setting developing new drugs, but also in frontline. And we need to rapidly move this innovation in the frontline of patients with high-risk tumor. And one of the high-risk tumors that we can consider is the IPG. So this Biomed program, which is up and running, is based on biopsy at diagnosis, looking at the expression of the target of 3 drugs, erlotinib, everolimus, and dasatinib, and proposing in this patient to receive on a randomly assigned way either erlotinib, everolimus, or the combination or the dasatinib, and generating through this randomized trial where things are being compared 2 by 2, a very smart design as well, that I will not comment anymore, but I can tell you it's a smart design, generating frontline in these newly diagnosed patients, adding new drugs to standard therapy, the capacity to provide access to innovation for these patients. So clearly, these 2 examples show that innovative design in the relapse setting and in frontline can really help to give more information in a way that we validate what we are doing. So in conclusion, what do we need to know about precision medicine? This is clearly for us not a demonstration that genomic medicine is better than chemo, but a way to develop better new drug for this patient in a well-characterized, molecularly characterized population. So clearly there is a need to access to innovative compounds in development, and this is still one of the challenges, to need to have a rapid access and early access to these drugs, and it needs to be done in partnership with pharma. Clearly, in the past, we have been developing drugs without pharma, but now we need to develop these drugs with pharma. And we need to consider, when we do academic trial, the possibility of having this data filed in a marketing authorization. And this is a new way to think about what we are doing, because partnering with pharma is a way to speed up, a way to access to innovation, but also to consider that what we are doing at some point, if it is effective, will need to lead to a marketing authorization to be sure that the drug is available for patients. And you tell me, but well, we are now in the field of immunotherapy. This is only for targeted therapy, and clearly we are well beyond. we have anti-JD2 treatment, we have CAR T cell, and we even see the PD-1 blockade in tumors with high mutation rate, including the large number of adult tumors responding to PD-1 and PD-L1. So is precision medicine just something that will disappear? I strongly believe that in immunotherapy as well, we will need information from the patient. We will need to follow what's happening to the tumor exposed to these drugs. So precision medicine and immunotherapy is not two different things, it's really linked together. And all what I told you, I think, is still valid if we consider development of innovative immunotherapy. So to conclude, I would like to thank all the people working on this program. They are young, they are creative, they are extremely smart. Just to tell you that this lady, Agnès Buzyn, was the president of the National Cancer Institute. She has been instrumental in developing the ISMART trial, and now she's health minister in France. And I can tell you, I'm very pleased to have a slide with the Minister of Health of my country to present to you. And I would like, of course, to thank the parents, the patients, and all the supports, the charities and the fundraising organization, and also the pharmaceutical company to make this happen, giving access to innovation for children, trying to do better for them. Thank you.